Hello viewers, I am Dr. Rovil. I work as a lecturer in pathology in a medical college hospital, and I am making this video for my students and also for you. Hope someone finds this helpful. Today's topic is on hemostasis. This video will contain the definition of hemostasis, followed by a brief overview on the various events of hemostasis. Then we will talk briefly about the roles of platelet, endothelium and coagulation cascade in hemostasis. Okay, so a lot of topics, so let's begin. First question, what do we mean by hemostasis? Now, the word hemostasis is derived from two ancient Greek words. Hem stands for blood and stasis means stopping or making something motionless. So, the literal meaning of hemostasis is therefore stoppage of bleeding or arrest of bleeding. And you also have to remember that three important things participate in this process of hemostasis, and those components are the platelet, the clotting factors, and the lining endothelium of our blood vessels. So these three are the major players involved in hemostasis. So how can we define hemostasis? So as written in your textbook, hemostasis can be defined as a precisely orchestrated process involving platelets, clotting factors, and endothelium that occurs at the site of vascular injury and culminates in the formation of blood clot, which serves to prevent or limit the extent of bleeding. Okay, I hope you're still with me. You didn't run away just like my students do when I try to teach them definitions of pathology. I even have to show them teddy bear to keep them calm. So look, I am also showing you a teddy bear to keep you calm. So look at the teddy, don't run away, because I will try to explain this definition line by line now. So, what did we see in the definition? In the first line we had said that hemostasis is a precisely orchestrated process. Now, have you ever been to an orchestral presentation? What do we see there? We see there are various instruments and all are played in a very well coordinated manner, in a very well choreographed fashion, so at the end they produce very beautiful music, right? Similarly, when we're talking about hemostasis, it is not just a random process that is happening whenever there is injury in a blood vessel. It is happening in a very well organized, well coordinated way. And that's why it is called a precisely orchestrated process. Now, who are the major players in this process? Like I said, they are the platelets, the clotting factor or the coagulation cascade, and the endothelium, the lining endothelium of our blood vessel. These three are the major player in the process of hemostasis. So, hemostasis can be defined as a precisely orchestrated process involving platelets, clotting factors, and endothelium. And where is this thing happening? It's not happening in an intact blood vessel. It's happening whenever there is injury to a blood vessel and blood is leaking out from those sites. That is the location. So it is occurring at the site of vascular injury and culminates in formation of blood clot. And why are we producing such blood clots at the site of vascular injury? The aim is to prevent or limit the extent of bleeding. So that was the definition of hemostasis. And this is a very important definition for your examination, so you can pause the video now to have a second look at the definition, because the examiners often assess whether students are reading the textbook or not by asking them definition. So no matter how much you hate definition, you have to remember definition, at least the important ones, to have good grades in your exam. So now that we have cleared the big definition, now we will move on to the next topic and talk about the sequence of events that are happening during hemostasis. Now always remember the sequence of events are the following. So initially there will be arteriolar vasoconstriction, followed by primary hemostasis, secondary hemostasis, and the last step will be clot stabilization and clot resorption. So now we will talk about these events one by one. So the first event of hemostasis is arteriolar vasoconstriction. Now always remember, whenever we are having damage in a blood vessel, blood is leaking through that damage. So our body will try to reduce the blood loss, and the best way to do that is to reduce blood flow to those arterioles, right? And that is the reason why we will see arteriolar vasoconstriction. Always remember that this is the first event of hemostasis. It will happen due to some neurogenic mechanism, and that neurogenic mechanism is augmented by endothelium. Now notice the term endothelium. As the name implies, this substance is derived from endothelium, and the function of endothelium is to act as a very powerful vasoconstrictor. So whenever there is damage in a blood vessel, the endothelium will release endothelin, and that will result in vasoconstriction, and vasoconstriction will reduce blood flow, and that will prevent further blood loss. So as we can see in the first image, so this is a diagram of blood vessel. Inside the lumen of the blood vessel we can see some cells, for example drawn in red. These are the red blood cells, and the tiny blue dots, these are the platelets. Okay, blood also contains white blood cells, but we haven't drawn those cells here to keep the diagram simple. Now notice what has happened here. So these are the lining of the blood vessel, right? So these cells, these are called endothelium, right? And you can see that the endothelium, inside them we have nucleus. Beneath the endothelium we can see that there is basement membrane. So this red thing is a basement membrane, and this is also basement membrane. And beneath the basement membrane, the dashed line are representing the smooth muscle cells. And we can also see that this part of the blood vessel has been damaged, and therefore the basement membrane and the subendothelial collagen are exposed. So what is happening during the first stage of hemostasis? The smooth muscles, due to the effect of endothelin that was derived from endothelium, will constrict. So the blood vessel will constrict. So the blood vessel will become narrower, and that will result in reduced blood flow. But also remember, this initial event is not permanent. It is a transient event, and if the subsequent stages of platelet activation and clotting factor activation, if those things doesn't happen, bleeding will resume again. So that was in short about the first stage. The second stage is primary hemostasis, and the main thing that is happening here is formation of platelet plug. Now how is that thing happening? So as we can see, the platelets will first adhere with the exposed collagen, and they will use von Willebrand factor as a cross-link between the collagen on one side and the platelet on the other side. And when we will talk about the role of platelet, we will discuss this mechanism in more details. So for the moment, just remember the second event is primary hemostasis. During this event, what is happening? First, the platelets will adhere with the exposed collagen, and they will do that with the help of von Willebrand factor. Now after adhesion, what will happen? The platelets will now become activated, And in your textbook you will see that platelet activation means two things. The first thing is alteration or change in the shape of the platelet. But then you will ask me, Dr. Robiul, what is the normal shape of the platelet? So always remember, the normal shape of the platelet is something like this: small, round, disc-like structure. So that shape will change into something like this once the platelets are activated. And in your textbook you will see a term that is sea urchin. Sea urchins are an animal that is seen in the deep sea, and they have a very interesting structure. You can Google that. I cannot show you the image of a sea urchin due to copyright reason, so you can Google about sea urchin and you will see that it is a very interesting animal. It has a spike surrounding it everywhere. Okay, so the platelet will have a shape that will have similarity with sea urchin, and I have also drawn that here. So during the stage of primary hemostasis, first there was platelet adhesion, that is followed by platelet activation, and platelet activation will have two things. One is the shape change that I just mentioned, and the second thing that will happen during platelet activation is secretion of different particles from the secretory granules of the platelet, and those secretory particles will attract additional platelets, that is known as recruitment, and then they will all aggregate together and form a platelet plug. So that is in short about the primary hemostasis. Moving on to the next stage of hemostasis, and that was obviously after primary we will have secondary hemostasis. Now what is happening during secondary hemostasis? So this is an image of secondary hemostasis. The main thing that will happen during secondary hemostasis is formation of fibrin meshwork. And how will that thing happen? Now always remember the subendothelial cells, that means the cells that are beneath the endothelium. So this is the endothelium, this is the subendothelial space. Similarly, these are the subendothelial space. And we have seen smooth muscle in subendothelial space. Sometimes there can be also fibroblast. So those subendothelial cells will release tissue factor. Tissue factor will combine with a clotting factor, that is factor 7, and that complex will ultimately result in formation of thrombin. And always remember, thrombin will cleave fibrinogen into fibrin, and fibrin meshwork will result in a very stable clot. So fibrin meshwork will be deposited over the primary clot or the primary platelet plug, and that will make that previously created primary platelet plug more stable. At the same time, thrombin has other effect on platelet, and that will also help in the stability of the plug. So these things will happen during the stage of secondary hemostasis. The last stage of hemostasis is clot stabilization and resorption. And actually during this stage there will be contraction of those clot that will make the clot more stable and permanent, and at the same time the counter-regulatory mechanism will also activate. For example, tissue plasminogen activator will begin to be liberated. And the main idea is to limit the clot only at the site of vascular injury. We don't want clot happening everywhere inside the blood vessel. Only in the damaged area we need blood clot. And those regulation will be achieved during this stage of clot stabilization, and ultimately there will be also resorption of clot. So these were in short about the various sequence of events or stages happening during hemostasis. So now we will move on to the next topic and talk about the roles of platelet, endothelium and coagulation cascade in hemostasis. So we will begin our discussion now with the roles of platelets. Now the major roles of platelet in hemostasis is formation of the primary platelet plug and also platelets provide a surface where clotting factors can bind and concentrate, as we will see. Now, platelets achieve these roles by a lot of components. For example, they involve some glycoprotein receptor that are located on the surface of the platelet. They also include the contractile cytoskeleton and two types of granules. Now this is very important. The examiners are very fond of asking the question that what are the types of granules that we can see inside the platelets. And always remember, there are two types of granules located inside the platelet. They are the alpha granules and the dense granules. Now, inside the alpha granules, what will we see? Well, on the membrane of the alpha granules, we will see P-selectin, that is an adhesion molecule. And inside the alpha granules, we will see some clotting factors, for example, fibrinogen, factor 5. We will also see von Willebrand factor and some factors, protein factors that are involved in tissue repair. For example, platelet-derived growth factor, transforming growth factor beta, fibronectin, and platelet factor 4. And in the dense granules of the platelet, we will see adenosine diphosphate, adenosine triphosphate, ionized calcium, epinephrine, and serotonin. And these informations are very important for your multiple choice questions. So now we know that the major role of platelet in hemostasis is formation of primary platelet plug and providing a surface where the clotting factors will bind and concentrate. And we have also mentioned various components of the platelet that help the platelet in achieving these roles. So let's now talk about the formation of primary platelet plug and discuss the role of platelet here. So like I said previously, first there will be platelet adhesion, and now let's talk about the glycoprotein receptors that are responsible for that adhesion. And in order to explain that, you can see that I have also drawn an image here. So this one is just zoomed in version of this portion of the blood vessel. So this is the basement membrane drawn in red color. This is the lining endothelium, and this portion of the endothelium has been damaged. So there is no endothelial cell here, and we can see subendothelial basement membrane is exposed, and drawn in blue here is depicting the collagen that is exposed. Now platelet will adhere with these exposed collagen, and in between the platelet and the collagen, von Willebrand factor will act as crosslink, and platelets will bind with von Willebrand factor using the glycoprotein receptor on the surface of the platelet, and that particular glycoprotein receptor is called GP1B. So this is the receptor. I hope you can see this. So this is the GP1B receptor as I have written here, and this GP1B receptor is binding with von Willebrand factor. Okay, and von Willebrand factor is bound with the collagen, so it is acting as a crosslink between the exposed collagen and the platelet, the GP1B glycoprotein receptor of the platelet. So that is adhesion. Now after adhesion there will be platelet activation, and we have already talked about the two components of platelet activation. Remember that one was the change in the shape of the platelet, and the other one was secretion of different types of components from the granules of the platelet. And we have also mentioned the shape. It has similarity with sea urchins. So that is the next step in formation of platelet plug, and that was platelet activation. Now always remember, whenever we are seeing the shape change that is followed by alteration in the glycoprotein receptor GP2B3A. So that's not the first receptor. The first receptor that we mentioned was GP1B, that was involved in platelet adhesion. But once the platelets have adhered and changed their shape, that shape change is followed by alteration in another glycoprotein receptor on the surface of the platelet, and that receptor is called GP2B3A. Now what will happen? These receptors will now have increased affinity for fibrinogen. So as you can see here, so these are the GP2B3A receptor. Here is one, and in between them we can see fibrinogen. So in a sense, fibrinogen is acting as cross-link between two adjacent platelets, and two adjacent platelets are binding with fibrinogen with the help of their receptor GP2B3A, and this thing is known as platelet aggregation. So there will be platelet aggregation, and ultimately that will result in formation of primary platelet plug. So these were the roles of platelet in forming the primary platelet plug, but also notice platelet had another role by providing a surface. Now how is that thing happening? Well, always remember, during the shape change, during the period of shape change, one interesting thing is happening, and that is some negatively charged phospholipid, for example phosphatidylserine. These negatively charged phospholipids are coming to the outer surface of the platelet, and there they will bind with calcium ion, and they will provide a surface where clotting factors can bind and concentrate. So that was the second role of platelet, by providing a negatively charged phospholipid surface where the clotting factors can bind and form complex. So these were, in short, the roles of platelet in hemostasis. Now one thing you have to remember, activated platelets will also produce thromboxane A2, and thromboxane A2 has a very important role in platelet aggregation as well. And also some diseases are associated with genetic deficiency of the glycoprotein receptors that I had just talked about, and also with deficiency of von Willebrand factor. So whenever there is deficiency of von Willebrand factor, that will result in a disease known as von Willebrand disease. Now I have a separate video on von Willebrand disease, and you can look into that video after finishing this for more information. Whenever there is genetic deficiency of the GP1B glycoprotein receptor, that will result in a genetic disease known as Soulier syndrome. And also whenever there is inherited deficiency of the other glycoprotein receptor, that means deficiency of GP2B3A glycoprotein receptor, that will result in a disease known as Glanzmann Thrombasthenia. So always keep these things in your mind for your exam. So remember, whenever there is deficiency of von Willebrand factor, that will cause von Willebrand disease. Genetic deficiency of GP1B receptor will result in Soulier syndrome, and genetic deficiency of GP2B3A glycoprotein receptor will result in Glanzmann Thrombasthenia. So now we will move on to the next topic and talk about the roles of endothelium in hemostasis. Now always remember, under normal condition, when the endothelium is not damaged and whenever the endothelium is intact, the antithrombotic functions of the endothelium will be predominant. Similarly, when the endothelium has been damaged, or whenever the endothelium is exposed to some pro-inflammatory factors, the antithrombotic properties of the endothelium will be lost and the prothrombotic properties will be predominant. So what are the antithrombotic function of endothelium? We can divide these under three broad headings. They are platelet inhibitory effect, anticoagulant effect, and fibrinolytic effect. So let's talk about these things one by one. So what are the anti or platelet inhibiting effect of endothelium? Well, the first one is very obvious. The endothelium will act as a barrier so that the platelets cannot come in contact with subendothelial collagen and von Willebrand factor. So that will prevent platelet adhesion and subsequent activation. Similarly, endothelial cells can release prostaglandin I2, nitric oxide, and adenosine diphosphatase, and all these things will prevent platelet aggregation. Always remember that we had said that ADP, adenosine diphosphate, is released from the dense granules of the platelet, and that thing had role in platelet aggregation. So whenever we're talking about an enzyme, that is adenosine diphosphatase, that can break this ADP, that will result in inhibitory effect on platelet aggregation. And also remember that endothelium has some molecules on its surface that can bind with thrombin and make the thrombin inactive. And we also knew that thrombin had a role in platelet activation and aggregation, so in a sense that will also inhibit platelet aggregation. So what are the anticoagulant effect of endothelium? Always remember, on the surface of the endothelium we have several types of molecules and they have anticoagulant properties. For example, there is thrombomodulin on the surface of endothelium that can bind with thrombin and that converts thrombin into an activator of protein C. Okay, so what is happening? Thrombin is binding with thrombomodulin, and then thrombomodulin is converting thrombin into activator of protein C. So ultimately protein C is getting activated, and also remember that this thing requires a cofactor, and that is protein S. So whenever there is formation of protein C, protein S complex, that will act as a very powerful anticoagulant, and this complex will inhibit factor 5A and 8A. On the surface of the endothelium we will also have heparin-like molecule that will activate antithrombin 3, and like the name implies, the activated antithrombin 3 will inhibit clotting factor thrombin, and it will also inhibit factor 9, 10, 11 and 12A. Similarly, we will also have tissue factor pathway inhibitor, and that will inhibit tissue factor as well as factor 7A. So all these were the anticoagulant activity of the endothelium. Regarding the fibrinolytic activity of endothelium, always remember that endothelium will liberate tissue plasminogen activator, and that will activate plasminogen and convert plasminogen into its active form, that is plasmin. And plasmin has fibrinolytic activity, that is, it can degrade the fibrins of the clot. So these were all the antithrombotic properties of endothelium. Now we will talk about the prothrombotic properties of endothelium. Now endothelial cells can liberate tissue factor or thromboplastin, and this thing will have a very important role in the extrinsic pathway of coagulation. Again, endothelium can also liberate platelet activating factor that will activate and also promote platelet aggregation. Endothelial cells can also liberate inhibitors of plasminogen activator, and they will act as antifibrinolytic agents. And also always remember that von Willebrand factor can be liberated from the endothelial cells as well, and we have already mentioned the role of von Willebrand factor in clot formation earlier. So these were the prothrombotic properties of endothelial cell. So we are almost at the end of today's discussion. The last thing that we will talk briefly is regarding the role of coagulation cascade in hemostasis. Now always remember, the coagulation cascade is a series of amplifying enzymatic reaction, and in these reaction the ultimate end product is fibrin. So this coagulation cascade will result in formation of fibrin, and that will have a very important role in clot formation. And one thing you have to remember, whether we're talking about the coagulation cascade that is happening in the test tube in the lab or in vivo, that means inside our body, some basic mechanisms are the same. For example, where is this reaction taking place? These reactions are taking place on the surface of phospholipid that was provided by the platelet. Recall that when we were talking about the role of platelet, one role was that a platelet can change its shape, and that shape change was followed by providing some phospholipid, some negatively charged phospholipid on the surface of the platelet, and this coagulation cascade is occurring on the surface of those phospholipid. And in these enzymatic reaction there is always a substrate that is usually the inactive form of the clotting factor, and there is also cofactor involved, and ultimately they are, once going through each step, they are becoming activated, and then they are again used in subsequent reaction. So on one corner of the board you can see this is the coagulation cascade that we see inside the lab. So it can be intrinsic pathway or it can occur through extrinsic pathway, and always remember that the extrinsic pathway is beginning with tissue factor. So tissue factor combines with factor 7, and they are making a complex of factor 7A. A stands for the active form, so factor 7A and tissue factor complex, and that will act on factor 10 and make the factor 10 active, and then factor 10A, the active form, together with factor 5A will act on prothrombin and convert the prothrombin into its active form, that is thrombin, and ultimately thrombin will act on fibrinogen and result in formation of fibrin. Similarly, whenever we are talking about the intrinsic pathway, it will begin with factor 12, and then we will see that it will activate factor 12 into 12A, that will act as enzyme for the next reaction, where we will have factor 11 becoming activated factor 11, and that will act on the subsequent reaction, and that will convert factor 9 into its active form, and together with the factor 9A and 8A they will again activate factor 10 into factor 10A. So it's a bit difficult to remember. The way I try to remember is I remember that the extrinsic pathway will begin with tissue factor, and the intrinsic pathway will begin with 12. And since 10 is 10 is common to both pathway, so I just put 10 in the middle and write down 12, 11 excluding 10, then 9 on one side, and tissue factor 7 on the other side to remember. Okay, so just you have to practice this thing more and more to remember this pathway. Moving on to the coagulation cascade that we see in our body, that means in vivo, we can see that that thing is beginning with tissue factor, that is activating factor 7 and converting that into factor 7A, and then factor 7 is acting on factor 9 and that is activating it and making factor 9A, and ultimately similar to the previous pathway, we are also seeing here formation of thrombin and ultimately formation of fibrin. So always remember that in the coagulation cascade, thrombin is very important, okay, because thrombin will have multiple function. One function of thrombin is to help in formation of fibrin. And what will be the role of fibrin? We have already talked about that fibrin first will be a monomer, then it will polymerize, and then it will act to stabilize the clot, and it will have also a role in formation of the secondary hemostatic plaque. So that is the role of fibrin. Again, thrombin will also amplify the function of factor 11, factor 8 and factor 5, and also thrombin, as you will see in your textbook, will have a role in platelet activation, and also it will have some pro-inflammatory roles as well. So these were in short about the role of coagulation cascade in hemostasis. So this concludes today's video on hemostasis. I hope this video was helpful. If you like my videos, do comment, share, subscribe and let me know. And for my students, I will also recommend you to go through your textbooks to know more information. Okay, so that's all for today. Until next time, take care and stay blessed. Thank you.